This is Jamaican sprinter Usain Bolt, and he's about to set the official world record for the fastest 100-meter sprint time.
There's the start, and Bolt got a good one, and Gay's right with him, and so's Asafa Powell. Bailey's there too, but here's Bolt. Bolt charging to the front. Gay's coming, but can't catch him. Usain Bolt, look at the time, 9:58, world record!
The title for the fastest man or woman on earth belongs to whoever owns the 100-meter sprint time. Why? Because it's the benchmark for all-out running speed. Elite sprinters look like they leave it all on track, but couldn't they eke it out a little bit more? In order to find out, we'll cover cellular respiration, including glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain, fermentation, and how to relate to dichloroacetate. Exhibit 1. Cellular energy. Cellular respiration is the biochemical process through which cells convert glucose and oxygen into adenosine triphosphate, or ATP, the energy currency of the cell, while producing carbon dioxide and water as byproducts. This is a catabolic reaction which breaks a larger molecule into smaller pieces, an exergonic reaction which releases energy as denoted by a negative Gibbs-free energy. There are two main types: aerobic respiration, which occurs in the presence of oxygen and involves glycolysis, the Krebs cycle, and electron transport chain to yield approximately 30 to 32 ATP molecules per glucose. Most eukaryotic organisms such as humans, animals, and plants primarily use aerobic respiration, which requires oxygen and efficiently produces ATP through glycolysis, Krebs cycle, and electron transport chain. And anaerobic respiration, which occurs without oxygen and includes processes like fermentation, yielding only 2 ATP molecules per glucose. This allows organisms to generate energy necessary for growth, movement, and cellular function. Anaerobic respiration is used in oxygen-depleted environments. Yeast, for example, performs alcoholic fermentation, converting sugars into ethanol and carbon dioxide. Along this line, ATP is a nucleic acid that serves as the primary energy carrier in cells. Its synthesis occurs through two mechanisms: substrate-level phosphorylation and oxidative phosphorylation. In substrate-level phosphorylation, a phosphate group is directly transferred from a high-energy substrate molecule to adenosine diphosphate, or ADP, forming ATP in the cytosol. However, it results in a small yield of ATP compared to oxidative phosphorylation. Oxidative phosphorylation is the process in the mitochondria in which ATP is produced driven by a gradient of protons across the inner mitochondrial membrane. In this process, electrons from carriers like NADH and FADH2 pass through the ETC, creating an electrochemical gradient by pumping protons from the mitochondrial matrix into the intermembrane space. The resulting gradient stores potential energy, which is then harnessed by ATP synthase as protons flow back into the matrix, producing ATP from ADP and inorganic phosphate. This chemiosmosis yields the majority of ATP in aerobic respiration. Electron carriers such as NAD+ and FAD are small organic molecules that transfer electrons from the breakdown of glucose to the ETC. Upon accepting electrons in hydrogen ion, NAD+ and FAD are reduced to NADH and FADH2 respectively. In redox reactions, one molecule loses electrons or is oxidized, while another gains electrons or is reduced. Now, cellular respiration itself is fundamentally a series of redox reactions characterized by the oxidation of glucose and a reduction of oxygen. During glycolysis, the Krebs cycle, and subsequent electron transport, glucose is oxidized, leading to the loss of hydrogen atoms and electrons, while oxygen is reduced as it accepts these electrons to form water. The mnemonic oil rig aptly summarizes this process. These are important for ATP synthesis as it couples the release of energy from electron transfers to the phosphorylation of ADP. Exhibit 2. Glycolysis. Glycolysis is a metabolic pathway that occurs in the cytosol or cytoplasm, breaking down glucose, a 6-carbon sugar, into two 3-carbon molecules called pyruvates, and releases energy in glucose to form ATP from ADP and inorganic phosphate, and NADH from NAD+. This process can be divided into two main phases: endergonic or energy requiring phase, and the exergonic or energy releasing phase. During the endergonic phase, two ATP molecules are consumed to phosphorylate glucose, transforming it into fructose 1,2,6,5-phosphate, which is unstable and easily splits into two 3-carbon sugars: dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or G3P. In the exergonic phase, each G3P molecule is further processed into pyruvates through a series of reactions that produce a total of 4 ATP and 2 NADH molecules for each glucose molecule. In the end, each molecule of glucose produces 2 pyruvate molecules along with a net gain of 2 ATP and 2 NADH. If oxygen is present, the NADH enters the electron transport chain to regenerate an NAD+ for glycolysis. If oxygen is absent, NADH is converted back into NAD+ through fermentation. Exhibit 3. Pyruvate oxidation. Pyruvate oxidation is an intermediate step in cellular respiration that prepares pyruvate molecules for the Krebs cycle. This process occurs in the mitochondrial matrix in eukaryotes and in the cytoplasm of prokaryotes, and starts with the transportation of pyruvate from the cytosol into the mitochondria. First reaction removes the carboxyl group from pyruvate, releasing it as carbon dioxide and leaving behind a 2-carbon acetyl group. This 2-carbon acetyl group is oxidized, and electrons are transformed to NAD+, forming NADH. Finally, the acetyl group is attached to coenzyme A, or CoA, creating acetyl-CoA, which is ready to enter the Krebs cycle. For each pyruvate molecule, one pyruvate is converted into one acetyl-CoA, releasing one carbon dioxide molecule and producing one NADH molecule. Exhibit 4. The Krebs cycle. Krebs cycle, also called the citric acid cycle, occurs in the mitochondrial matrix of eukaryotic cells. First, acetyl-CoA, a 2-carbon molecule, combines with oxaloacetate, 4-carbon molecule, forms citrate, 6-carbon molecule. Citrate is then rearranged into an isomer called isocitrate. The first two oxidative decarboxylations which is a chemical reaction that removes a carboxyl group and releases carbon dioxide. As a result, happen next. Isocitrate is oxidized and loses a carbon as carbon dioxide, reducing NAD+ to NADH, producing a 5-carbon molecule. This undergoes another oxidation, losing another carbon as carbon dioxide and reducing another NAD+ to NADH. This reaction also attaches to CoA, forming succinyl-CoA, a 4-carbon molecule. The CoA group in succinyl-CoA is replaced by a phosphate group, transferred to ADP to form either ATP or GTP, depending on the cell type, yielding succinate. Another 4-carbon molecule, succinate, is then oxidized to fumarate, and in this step, FAD is reduced to FADH2 because 2 hydrogen atoms are transferred. Water is added to the 4-carbon molecule, converting it into another 4-carbon molecule, which undergoes another oxidation to produce oxalose acetate, which is our starting 4-carbon compound. Another molecule of NAD+ is reduced to NADH in the process. Each full turn of the Krebs cycle yields 3 NADH, 1 FADH2, and 1 ATP or GTP, as well as releasing 2 molecules of CO2. Additionally, oxaloacetate is regenerated at the end of each cycle, allowing it to react again with a new acetyl-CoA molecule. Because each glucose molecule produces 2 pyruvate molecules and thus 2 acetyl-CoA molecules, the cycle turns twice per glucose molecule, yielding a total of 6 NADH, 2 FADH2, and 2 ATP or GTP molecules, along with 4 molecules of CO2. Exhibit 5. The electron transport chain. The electron transport chain, or ETC, is the final stage of cellular respiration, occurring specifically in the inner mitochondrial membrane. It consists of a series of protein complexes, labeled 1 through 4, and electron carriers that facilitate electron transfer. Electrons are supplied by NADH and FADH, generated in glycolysis, pyruvate oxidation, and the Krebs cycle. NADH donates electrons at complex 1, while FADH2 enters at complex 2. These electrons move sequentially through the complexes, releasing energy through a series of redox reactions. This energy powers complexes 1, 3, and 4, pump electrons, or H+ ions, across the inner mitochondrial membrane from the matrix to the inner membrane space, creating a high proton concentration outside the matrix. This proton gradient, or proton motive force, drives protons back into the matrix through ATP synthase, a protein that uses this movement to synthesize ATP from ADP and inorganic phosphate. Without this protein gradient, ATP synthase would lack the energy to efficiently produce ATP. At the end of the ETC, electrons combine with oxygen, the final electron acceptor, and protons to form water. Electrons are transferred to molecular oxygen or O2 at complex 4, where they combine with protons from the matrix to form water as a byproduct. For each molecule of glucose, the ETC can produce approximately 26 to 28 ATP molecules, along with water and regenerated NAD+ and FAD. Exhibit 6. Fermentation. Lactic acid fermentation occurs in the cytoplasm of cells when oxygen levels are insufficient for aerobic respiration, allowing organisms to regenerate NAD+ from NADH produced during glycolysis. This process begins with glycolysis, where 1 glucose molecule is converted into 2 pyruvate molecules, netting 2 ATP and 2 NADH in the process. In the next step, NADH donates its electrons directly to pyruvate, resulting in the formation of lactate and the regeneration of NAD+, which provides a constant supply of energy under anaerobic conditions. Lactic acid fermentation is particularly relevant in human muscle cells during intense exercise when oxygen is scarce, leading to temporary lactate accumulation. This lactate can be processed back into pyruvate in the liver for further energy production. Alcohol fermentation, which allows yeast to produce ATP in the absence of oxygen, also takes place in the cytoplasm and follows a similar initial step of glycolysis, producing 2 pyruvate molecules, 2 ATP, and 2 NADH. However, this process diverges from lactic acid fermentation at the pyruvate stage. First, each pyruvate undergoes decarboxylation, releasing carbon dioxide and forming acetaldehyde. Next, NADH donates its electrons, regenerating NAD+ and producing ethanol as a byproduct. This pathway allows yeast to produce ATP anaerobically by converting sugars into ethanol, and is used to produce alcohol in brewing and winemaking. In this way, both fermentation processes allow organisms to extract energy from glucose when oxygen is unavailable. Exhibit 7. Applications of cellular respiration. Dichloroacetate, or DCA, is a compound originally developed to treat metabolic disorders, particularly congenital lactic acidosis. Functions as a metabolic modulator by allosterically inhibiting pyruvate dehydrogenase kinase, or PDK, an enzyme that negatively regulates the conversion of pyruvate to acetyl-CoA. When PDK is active, it phosphorylates and inhibits the pyruvate dehydrogenase complex, or PDC, reducing pyruvate conversion to acetyl-CoA and diverting it towards lactate production. By blocking PDK, DCA activates PDC, allowing more pyruvate to enter the mitochondria, where it is converted to acetyl-CoA and funneled into the Krebs cycle for further oxidation. This action promotes aerobic metabolism and decreases lactic acid accumulation, making DCA a focus of cancer therapy research, as many tumors exhibit altered cellular respiration, called the Warburg effect, and favor anaerobic pathways, even in the presence of oxygen. DCA specifically targets the pyruvate oxidation step in aerobic respiration. Under normal conditions, pyruvate produced during glycolysis is converted to acetyl-CoA by the PDC. However, in cells with active PDK, PDC is inhibited, causing pyruvate to divert toward lactate production. DCA alleviates this inhibition by inactivating PDK, restoring PDC activity and thus enhancing the flow of pyruvate into the Krebs cycle. This promotes efficient ATP production through oxidative phosphorylation rather than lactate production, which counters the tendency of cells to rely on anaerobic metabolism. By increasing acetyl-CoA availability, DCA enhances the Krebs cycle and ETC. leading to higher ATP output and reduced lactate accumulation. To mitigate the impact of DCA on pyruvate oxidation and its associated side effects, treatments focus on balancing the mitochondrial response to DCA-induced oxidative stress. For example, antioxidants like N-acetylcytosine or NAC or alpha-lipoic acid are administered to specifically counteract the excess reactive oxygen species or ROS generated during the DCA-activated pyruvate oxidation process. These antioxidants help preserve mitochondrial integrity by neutralizing ROS, which prevents oxidative damage to enzymes in the PDC and maintains efficient conversion of pyruvate to acetyl-CoA. By stabilizing the pyruvate oxidation pathway, these treatments allow for sustained ATP production through oxidative phosphorylation without excessive ROS buildup. Exhibit 8. Conclusion. So why is it almost impossible for humans to run a 100-meter dash in 9 seconds flat? Well, due to limitations of human cellular respiration, particularly the transition between anaerobic and aerobic energy systems. During such a short and intense effort, the body primarily relies on anaerobic pathways, particularly glycolysis and lactic acid fermentation, which rapidly converts glucose to pyruvate, yielding a net gain of 2 ATP molecules. However, this process quickly leads to the accumulation of lactic acid when oxygen is scarce, resulting in muscle fatigue. For sustained performance, aerobic respiration through the Krebs cycle and ETC is essential because it produces significantly more ATP, approximately 30 to 32 ATP per glucose. It operates too slowly to meet the immediate energy demands of sprinting. During a brief 9-second 100-meter sprint, the body cannot efficiently transition from anaerobic pathways, like glycolysis and lactic acid fermentation, to aerobic respiration. As a result, it struggles to produce ATP quickly enough to sustain such a high-intensity effort.